Friends, in a previous lecture we discussed about habit and habitats, morphology, digestive system and excretory system. Now, in this episode we will discuss about the nervous system, reproductive system, life cycle and the infections of liver flukes. Now, let us start with the nervous system of liver fluke. Please look at the figure. You can see the nervous system which is mainly situated around the pharyngeal region. Generally, the sensory organs which are present in the animals are lacking in liver fluke because the animal is an endoparasitic animal. Now, look at the figure again. Surrounding the pharynx, a pair of lateral ganglion and the ventral ganglion connect to the form cerebral ring. You can observe very clearly the ring which is situated surrounding the pharyngeal region. The three pairs of longitudinal cords run backward and connected through the commissure, and the interior part having fine nerves are arising through the brain. So, by this nerves and this commissure and this ganglion, almost all nervous systems are getting completed and which connect almost all the parts of the body. Now, friends, let us discuss about the reproductive system. It is very precious and very important system or very important part of the life of the liver fluke, because through this part it can reproduce and it can generate a new liver fluke for the new infections. Now, look at the figure please. You can see a very dense figure over here having a numerous of flame cell including the reproductive system. Now, friends, let me tell you about the reproductive system of the liver fluke. This is the very lower kind of invertebrate. Just like almost all invertebrate, it also known as a hermaphrodite animal. I think you all know that hermaphrodite animals means the male reproductive parts as well the female reproductive organs are present in a single animal, and both the male and the female reproductive organ will produce the spermatozoa as well as ova. In this animal, the self cross or the self fertilization is very rare. Self fertilization, self cross means that the own spermatozoa will fertilize the own ova. It is very rare activity in the liver fluke, but they are showing the cross fertilization. It means that the spermatozoa of one animal and the ova of the another animal. Now, let us discuss about the male reproductive system first. Look at the figure, friends. You can observe the different male reproductive organ. We will start with the testes. These are very greatly ramified and tubular structure situated in the middle part of the body, one behind the another. You can see in the figure it is very reticulated part which is situated at the exactly middle part of the animal, and it is named as the anterior testis as well as the posterior testis. You can see in the label very clearly. From the testis, the another organ we will discuss, that is vas deferentia. The vasa deferentia is a narrow delicate duct. The vasa deferentia emerge out from the each testis and reams forward to meet the fellow of opposite side, forming the common sperm duct just below the ventral sucker. Now, friends, you can see situation of the ventral sucker and they are getting merged with each other in the figure. Afterward, The another organ is seminal vesicle. It is also known as seminal vesicle. The common sperm duct immediately leads into a large muscular pear shaped sac. Please observe the figure, friends. This seminal vesicle, which lies in the front of ventral sucker, it is very clear, you can observe in the figure. Just like this, all the male reproductive organ collectively form the male reproductive system. Now, let us see in slightly enlarged figure. You can see in the enlarged figure that male genital aperture, female genital pore and genital atrium. Now, during the breeding season, the male reproductive organ will develop a cirrus like structure and cirrus sac. You can see in the enlarged figure, it is also known as penis of the liver fluke. You can see the ejaculatory duct which is entering into this cirrus, which is coming in the zigzag from the seminal vesicle. The cirrus to open into the genital chamber through the male genital pore. The cirrus is the muscular and the cylindrical structure. You can see in the figure, and it looks just like which I am describing. The cirrus or the penis is a muscular cylindrical structure transverse internally by the passage of ejaculatory duct. You can see in the figure very clearly what I am describing. It can be pushed out and drawn into through the genital pore and thus help in the copulation. The cirrus and the seminal vesicle both are enclosed in a bag like cirrus sac. See in the figure that genital atrium and the cirrus which is coming out from the genital aperture. Now, the prostate gland, you can see many unicellular glands which is situated surround the ejaculatory duct. They are very numerous and which secrete the alkaline secretion which help the free movement of sperms during the copulation. Now, friends, let us discuss about the female reproductive organs. Look at the figure again, friends. This is the figure of the smaller region of the part of the female reproductive organ. This figure will show the small part of the ovary which will open into the oviduct, which will open into the yolk reservoir and the followed by the uterus. Now, friends, you can also observe that there is a opening of the lower canal. See, during the morphological description, we did not mention anything about the lower canal. Why? Because this lower canals will produce only during the reproductive season, afterward it will disappear. So, please look at the lower canal. Now, let us discuss the female reproductive organ one by one. The ovary, it is a very primary organ of the female reproductive organ. It is also known as germarium, which is very large, highly branched, tubular, which is situated on the right side in the front of the testis. You can see the previous figure that testis and ovary both are situated together, but in the right side the ovary is situated, which is short, narrow and convoluted, that is oviduct. It is also known as the vitelline duct and runs downwards to join the uterus. I think you are observing where the uterus is situated. Now, let us discuss about the uterus. It is very long, which you can see in the figure, and highly convoluted tube that extend up to the genital atrium opening into it through the female genital aperture and close to male genital pore on the left side. You can observe in the figure, you can see the enlarged figure that in the genital aperture both the male and female genital apertures are opening side by side. It contains a large number of capsules which have fertilized eggs. Now, vital area gland. It is one of the important gland associated with female reproductive system. The junction of the median and transverse vitelline duct swells up and form a yolk reservoir. The yolk reservoir will help for the development of the embryo. Another gland is Mealy's gland. Please look at the figure. Mealy's gland is a tough and also known as shell gland, which do not play any role in the shell formation. It remains surrounding the base of the oviduct and its secretion help in the lubricating uterus for the smooth movement of the eggs and sperm. Now, friends, let us discuss about the Leurer's canal, which produce only during the breeding season. This canal is arising from the oviduct during the breeding season, and it is also acting as a sperm duct. Sperm duct means the spermatozoa are getting entered into the female reproductive organ through this canal. So, it is also known as the vagina of the animal. So, like this we just discussed about the male and female reproductive organ. Now, how this organ works, how the copulation takes place, how the fertilization is taking place, and how the life cycle is get started. Let us discuss about this. Now, friends, look at the life cycle. By the first sight, you can observe that we have drawn two animals. Number 1 at the above, that is sheep, and number 2 at the below side, that is snail. It means that we have drawn two animal, and we have already discussed that the life cycle which taking place into two hosts. That is why this life cycle is known as digenetic life cycle. Again I am telling, because of it covers two hosts, that is why this life cycle is known as digenetic life cycle. Now, look at the figure, friends, again how the copulation is taking place. Now, please try to recall the previous figure, that during the cross fertilization, let me tell you again, the cross fertilization means the two fasciola, it means the two liver flukes are taking part in the copulation, and the spermatozoa of the one animal will enter into the another animal is known as cross fertilization. Generally, the higher vertebrates are showing this kind of fertilization or copulation. Now, how the formation of capsule is taking place? Now, look at these four figures. You can see the figure number 1, the fertilized egg, it is also known as zygote, which covers by the 30 to 50 yolk cells and the shell globules from the vitelline gland. By the way of various vitelline duct, the shell is get formed with the lead or with the operculum at one side, generally at the interior side. You can observe the another figure. The zygote and the mass of yolk cells enveloped by the shell constitute the capsule. When a complete capsule passes into the uterus, the capsule formation occurs throughout the year, and the one fluke may produce more than 50,000 capsules in a year. Now, look at the another figure, which is also known as the embryonic development. Now, what is embryonic development? After the fertilization, the ova is known as zygote, and the development of zygote leads to the adult animal. that is known as the embryonic development. Now friends, look at the another figure. The cleavage start immediately after the fertilization within the capsule, and the capsule is situated in the uterus and which contain the mass of the embryonic cell. The encapsulated embryos, or it is also known as capsule, do not develop further in the fluke's uterus. So, what is happening? The capsule then escape through the gonopore and are liberated the bile capillaries, from where they reach the intestine to the host, and ultimately with the fecal matter it will get out of the host body. Now, you can see the figure of the life cycle. Whenever the embryos will come out through the fecal matter, it will survive for a few months with the wet fecal matter without any development, and the further development occurs when the capsule come into the contact with water in the optimum temperature of the 30 degrees Celsius. The encapsulated embryo differentiated into Miracidium larva. You can also observe in the fourth figure of the capsule that Miracidium larva is getting developed inside the capsule. You can observe the figure of the Miracidium larva. It is another stage of the life cycle which is taking place outside the host body. Now, see the figure please. It is the very first larval stage, the encapsulated embryo in 4 to 15 days, which will differentiated into Miracidium larva. It will hatch out and swims in water, and the speed of the swimming of this larva is very less. Now, look at the figure friends. It is very minute and 0.07 millimeter long. It is oval, elongated and richly ciliated. You can observe the many cilia which is surrounding at the periphery of the larva. This is the multicellular organism. Within the Miracidium body, there are glands, nervous tissue, protonephridia and germ cell. You can see the label which is situated aside of the figure. Miracidium does not feed. During the swimming, Miracidia tries to penetrate any object or organism, but only organism which is the another host of the animal, that is limnia. If the Miracidium will not able to come into the contact of limnia, it will die within 24 hours. But when it come into the contact of the limnia, it will enter into the host body by the boring mechanism. You can see in the figure how the Miracidium larva are entering into the body by the boring mechanism. You can see there are 1, 2, 3, 4, 5, 6 figure. The first figure they are coming into the contact. Number 2, the apical papilla of the Miracidium is penetrating inside the epithelial. Then slowly step by step the whole Miracidium larva is getting entered into the snail body. Afterward, it will reach to the digestive gland of the snail and it will change into its body in 14 days and developed into the another larva, that is known as Sporocyst larva. Look at the figure of the Sporocyst. It is also a miniature creature, and you can also observe that small another larvae are situated inside the Sporocyst. This Sporocyst is a elongated hollow sac like larvae, and its body wall consists of non-cellular cuticle. The brood chamber is a hollow space, and in the center of the sporocyst which are found the germ balls. Now friends, what is germ ball? Germ ball is a group of germ cell. So, many germ cells will come together and form a ball like structure and form a germ balls. You can also see in the figure. This sporocyst moves about the muscular contraction in the host tissue that absorb the nourishment from them and causing them the considerable damage. So, due to this, the snail, that is the host, will get damaged. Its germ ball develop into the next larval form, that is known as radia. Each sporocyst produce about 5 to 8 radia in it. You can observe in the figure. Now friends, look at the another figure of the radia larva. In this larva, you can observe a very acute variation, that is the formation of mouth and pharynx. You can see this larva is very active and very small creature, which nourishes by the sucking fluid and the cells of the host tissue by the muscular pharynx. Now friends, we already discussed about the muscular pharynx. So, the movement of muscular pharynx, or we can say the function of the muscular pharynx, will start right from the radia larva. Immediately this larva will migrate into the digestive gland of the snail. During the summer month, now please try to concentrate on the season. During the summer month, when the food is sufficiently available, the mother radii mature in 12 to 18 days. The germ balls gives rise to a second generation of the daughter radii. These germ balls of the daughter radii in the winter will develop into next larval stage, that is known as Cercaria, that you can observe in the figure. This is the slightly different kind of larva, and you can observe all the parts which found in the adult liver fluke. It also contain a tail like structure for the movement. This larva, that is Cercaria larva, will come out from the radia through the birth pores. You can observe in the figure, birth pores are situated from the either side of the larva. Almost 14 to 20 Cercaria larvae are getting produced, and they are 0.25 to 0.35 millimeter long. They are all and containing tail. You can see in the figure that the interior part contain the oral sucker, which is also situated in the adult liver fluke, and mouth with a sucker and the suctorial pharynx. It contains long esophagus, which will be shorter into the adult liver fluke. Then the blind intestine. The ventral sucker, or it is also known as acetabulum, is present on the ventral side. From the digestive gland of the snail, the Cercaria passes into the pulmonary sac, and then it will escape from the host body in the surrounding water. Cercaria swims up to 5 minutes to an hour in the surrounding water. Afterward, it will settle down on some green leaves of a water plant that you can observe in the figure of the life cycle. Later on, tail is cast off. Cast off means it will removed. The body becomes rounded and forms around itself a thick brownish cyst wall from the cystogenous gland. The encysted Cercaria is now known as metacercaria. This is the last larvae or last stage of the life cycle of the liver fluke. Friends, look at the figure. It is rounded, and you can also observe that a thick cyst wall is situated around it. It is a juvenile fluke having a rounded and with a diameter of 0.2 millimeter. It resembles Cercaria larva. The metacercaria insisted on vegetation and may live for as long as a year at a low temperature and for as a short time as 2 or 3 weeks at 25 degrees Celsius. Now, friends, how the infection is carried out by the larvae or by the liver fluke in the primary host. Now, look at the life cycle, that how infection is taking place. Metacercaria develops into adult fluke only inside the definitive host or a sheep. The sheep get infected during a grazing on leaves and a grass to which metacercaria cysts are attached. Afterward, please look at the figure. Metacercaria survive from action of the host gastric juice. This covering of the metacercaria will help to provide protection from the gastric juice of the host, which penetrate into the wall of intestine and enters into the coelomic cavity. Coelomic cavity, I think, friends, you all know that this is the cavity which lies between the digestive tract, which is alimentary canal, and the body wall. From the coelomic cavity, it swims into the coelomic fluid and enter into the liver, or in other words, we can say it infect the liver. It will feed on the liver tissue and grow in the size of 5 to 6 weeks, and it take a shelter in the bile duct, where it finally attained a sexual maturity in 11 to 13 weeks. This metacercaria will get developed and converts into a mature liver fluke, which reside in the bile duct. Friends, up to the point we just discussed about the different system and the life cycle of the liver fluke. Now, let us discuss about the pathogenesis of the liver fluke. It means that how the liver fluke is injurious to the health of sheep and human beings. Now, look at the figure. Whenever the metacercaria will enter into the host body and it will convert into the adult body, may be either chronic or acute. This is known as acute fasciolasis. During the acute fasciolasis, the liver root occur during the pre-adult migration of the fluke in the parenchyma of the liver and sometime the other organs also for about the 8 weeks. During the infection, the liver becomes swollen and the byproduct of the liver fluke are very toxic, which will highly damage to the liver. The chronic fasciolasis occur beyond the 12 weeks of infection, and when the flukes have reached the bile ducts and are maturing sexually, it will affect adversely on the production of milk and wool, and sometime it leads to the death of the host also. Now, how we can save the host from the acute infection of the liver fluke? So, please look at the chart. We can give a different treatment to the host or a sheep by giving different medicines, just like carbon tetrachloride, hexachlorothene, bithionol, praziquantel, and triclabendazole. These are some medicines which can be given to the sheep after the consult with the veterinarian or the expert person, and we can get a cure from the fasciolasis. Friends, in this lecture, we learn about the nervous system, reproductive system, life cycle and pathogenesis of the liver fluke. So, combinedly in 2 lectures, we discuss all the system and pathogenesis of the liver fluke, and I hope that you thoroughly understand it. Thank you.